So hi everyone. Today I'm going to discuss about a project we developed at Roche, which is building a causal gene network in inflammatory bowel disease using Bayesian modeling of omics data. My name is Chaimae El Houjjaji, and I'm working in the Immunology, Infectious Disease and Ophthalmology department in Roche Pharmaceutical Research and Early Development period. So we will first give a quick introduction about the disease before explaining the algorithm, methods, and finally give some results that we got from our network. So first, inflammatory bowel disease, IBD, is a chronic autoinflammatory disorder that affects the digestive system caused by an abnormal immune response to the intestinal microbiome. It is characterized by inflammation of the digestive tract and results from a disruption in the balance between genetic, immune, environmental, and microbiome factors. So IBD typically affects the colon and the small intestine, but can affect any part of the digestive tract. And there are two main types of IBDs, so Crohn's disease, CD, and ulcerative colitis. And so here there are different types of biologics used from what we can see on the therapeutic pyramid, but the range of response to the existing biologics is only of 30 to 50% depending on the patients. So our aim is to raise this efficacy ceiling, and there's a urgent need nowadays to identify novel targets and advance understanding of the key drivers of IBD. In order to help doing that, we built a causal gene network in UC based on genomic, transcriptomic, and clinical data. These probabilistic causal network models can elucidate the regulatory architecture of UC, leading to the identification of key driver genes, which can modulate the disease. So we run our Bayesian network using two gene expression datasets. So the first one is etrolizumab dataset, which is a Genentech Roche UC data, which is composed of 661 patients that were treated either with anti-integrin beta-7, anti-TNF, or placebo. And we have more than 2,300 genes as an input to the network for these datasets. The other dataset that we applied our algorithm on is a public UC dataset from the Mount Sinai Hospital, composed as well of 661 patients, with biopsies from inflamed and non-inflamed regions, and we have more than 3,000 input genes in our network for these datasets. So the approach that we use is the following one. So we create an initial list of input genes composed of differential expressed genes in each of the datasets that we presented. We add to these genes that we extracted from co-expressed models by applying WGCNA on the two gene expression datasets that we showed before, and we also add GWAS genes that we gathered from multiple literature studies. On top of that, we add some prior knowledge, such as information from ccQTL, transcription factor information, so linking transcription factors and their target genes, as well as protein-protein interaction edges. We use then the computational tool RIMBANET, that stands for Reconstructing Integrative Molecular Bayesian Network, which will output a consensus network out of 1,000 Bayesian network run on the dataset that we gave him. And then we have as an output a directed and weighted causal gene network that we can use in order to identify key driver genes, new gene-gene interaction, but also make a visualization app that we developed in order to help for target identification. So this causal gene network can be used for multiple applications. For example, we can use it for exploring gene neighborhood of relevant but undruggable targets. We can also use it for evaluating the overlap of subnetworks around specific genes that could be targeted through B-specific approaches. So in the figure that you can see on the right, we gave him the example of two input genes in yellow. We can see their neighborhood, And we can also see the overlapping of their neighborhoods, which are the bee-colored genes that we can see in the middle of the figure. And finally, one third application could be predicting key driver genes whose expression affects disease-associated pathways that we will develop in the next slide. And the network visualization and exploration tool was also developed on Chaimae app in order to help in targeted identification and specific applications that we gave as an example before. Moreover, weighted key driver analysis, so WKDA, was applied as well to identify hub gene nodes, so that we call key driver gene nodes, and pathway enrichment of the key driver gene subnetworks revealed central disease processes. Some of the top 10 most enriched pathways that we showed on the poster have been previously associated with IBD, such as TCR signaling, B-cell signaling, which is confirming the utility of this approach. And finally, the key driver gene subnetwork analysis uncovered some previously known regulators of inflammation, such as CASP8 that we can see in the figure on the right, but also STAT33 that is also present in the same figure, but also genes which can represent potential new targets for IBD in the future. So to conclude, we demonstrated that the causal network built using a Bayesian approach based on transcriptomics, genetics, protein-protein interaction, transcription factors, and ccQTL data sources reveal key disease processes in UC. And the identified regulatory genes driving these processes have been previously associated with IBD or can be used for further experimental validation to help identify therapeutic targets. Thank you.